Hello friends, welcome to bioassembling. In this session, I will discuss about the detailed structure of prokaryotic cell. Let's start the session. Prokaryotic cell which contain prokaryotic cell which means before nucleus in Greek. They include all cells which lack nucleus and other membrane bound organelles. Means they do not contain any membrane bound organelles. Examples of prokaryotic cell are mycoplasma virus, bacteria, cyanobacteria, or blue-green algae. Most prokaryote size range from 1 micrometer to 10 micrometer, but they can vary in size from 0.2 micrometer to 750 micrometer. Example is Margarita nambiensis. They belong to two taxonomic domains, which are the bacteria and the archaea. They belong to two taxonomic domains, which are bacteria and archaea. Most prokaryotes are unicellular, except myxobacteria, which have multicellular stage in their life cycles. They are membrane-bound, mostly unicellular organisms, lacking any membrane-bound organelles. Means unicellular organisms do not contain any internal membrane-bound organelles. These are the typical prokaryotic cell structure. These inner membrane contain nucleoid or circular DNA. Means prokaryotic cell do not contain any nuclear membrane. DNA present inside the cytoplasm in the form of circular structure, and these are cytoplasm fluids present inside the plasma membrane of prokaryotic cell. These are the cell wall. Cell wall is present middle side of the plasma membrane and capsule. Capsule are the outer structure of plasma membrane which provides shape of for prokaryotic cell. Means capsule provides shape of prokaryotic cell like bacteria, etc. And this you can see that green structure is called bacterial flagellum. Bacterial flagellum helps cell to for locomotion, and it also act as a sensory organ, which means it sense any chemical compound present outside of the prokaryotic cell. It can sense it. These are the ribosome, small teeny molecules. You can see this. These are ribosome, which ribosome help in protein synthesis with the help of messenger RNA. And this is plasmid. Plasmid are extra chromosomal circular DNA which can replicate itself. And you can see this rod-like structure, green rod-like structure, which are embedded in the prokaryotic cell is called pili. And these all are the structure of prokaryotic cell. Here is the detailed component of a prokaryotic cell like flagella. It is a long whip-like protrusion found in most prokaryotes that aids in cellular locomotion. Besides its main function, locomotion is also function as a sensory organelle, being sensitive to chemical and temperature outside of the cell, which means flagella is long whip-like protrusion just which help in cellular locomotion. Is I- Besides its function, locomotion is sensory organelles, and it sense chemicals and temperature. Capsule. Capsule is found in some bacterial cell, and this additional outer covering protect the cell when is engulfed by phagocytosis and phagocytes and by viruses. When viruses are engulfed with bacteria, then capsule help the bacteria to protect us. And the capsule retain moisture and helps the cell adhere to surface and nutrients. The capsule is found in most commonly among gram-negative bacteria E. coli, Klebsiella pneumonia, Haemophilus influenza, Pseudomonas aeruginosa, and Salmonella are the some example of gram-negative bacteria which contain capsule. There is the Example of gram-positive bacteria are Bacillus megateriens, Streptococcus pneumonia, Streptococcus pyogenes. Next, cell wall. Cell wall is outermost layer of the cell which protects the bacterial cell and give it shape. One exception is mycoplasm which lacks cell wall, which means mycoplasm do not contain any cell wall. Bacterial cell walls are made up of peptidoglycan which is made from polysaccharide chain cross-linked in usual peptides containing D amino acids. Means D amino acid is cross-linked between polysaccharides and form peptidoglycan layer. The cell wall of are different from this the plants and fungi which are made up of cellulose and chitin respectively. Plant cell wall and fungi cell wall are made up of chitin and cellulose, and bacteria cell wall is different from plant cell wall. Cell wall of bacteria is also distinct from the archaea which do not contain peptidoglycan. Archaea do not contain peptidoglycan, so the cell wall of bacteria and archaea differ. On the basis of gram staining, bacteria is divided into two types, gram positive and gram negative. First of all, the name originates from the reaction of cells on gram stain, a test long employed for the classification of bacterial species. And gram positive bacteria possess a thick cell wall containing many layers of peptidoglycan and teichoic acid. In contrast, gram-negative bacteria have a relatively thin cell wall consisting of few layers of peptidoglycan surrounded by second lipid membrane containing lipopolysaccharide and lipoprotein. I will repeat: gram-positive bacteria thick cell wall and gram-negative bacteria contain thin cell wall. The thick cell wall is because of gram-positive bacteria contain layer of peptidoglycan and teichoic acid, and gram-negative bacteria do not contain the layers of peptidoglycan. Few only contain few layers of peptidoglycan surrounded by second lipid membrane containing lipopolysaccharide and lipoprotein. And if this difference in structure can produce differ in property of antibiotic susceptibility. For example, vancomycin kill only gram-positive bacteria in is infective against gram-negative pathogen. For example, vancomycin can kill only gram-positive bacteria. It do not kill gram-negative bacteria because gram-negative bacteria resistance to vancomycin, and such as Pseudomonas aeruginosa and Haemophilus influenza are the example. Here is all about the gram positive and gram negative. Next page I will discuss about the structure of gram positive and gram negative bacteria. These are the cell wall of gram positive bacteria and these are the cell wall of gram negative bacteria. The cell wall of gram positive bacteria contain thick cell peptidoglycan layer, whereas gram-negative bacteria contain thin peptidoglycan layer. You can see that the peptidoglycan layer present in the gram-positive bacteria is higher. Many layers are present in gram-positive bacteria, whereas in gram-negative bacteria only two layers are present. And outside of the gram-negative bacteria, outer membrane is lipopolysaccharides or lipids membrane are present. Outer surface contain proteins, molecules and porins which are permeable to small molecules, and these are lipids and lipid A or lipopolysaccharide, and which contain O polysaccharides. These are O polysaccharides, and these small molecules you can see that is a lipoprotein, and these all are absent in gram positive cell wall. Gram positive cell wall contain only surface protein, teichoic acid and lipoteichoic acid chain. And similar things is both of gram positive and gram negative bacteria is the inner side membrane or cytoplasmic membrane which are similar. You can see this, and phospholipids are present both, and these all about the gram negative and gram positive cell wall. You can see that the Morphological structure of prokaryotic cell. First of all, it is coccus. Its spiral oval shape is called coccus, and it is streptococcus. Means when coccus is formed, chain-like structure is called streptococcus. This is bacillus, single bacillus, and this is streptobacillus. Means when bacillus is formed, chain-like structure is called streptobacillus. And this structure is spiral shaped, is called spirochetes, and this is vibrio. And you can see that single flagella is present in this bacteria, and is that multiple flagella are present. And these all are the structure of prokaryotic cell. Reproduction. Bacteria and archaea reproduce through asexual reproduction known as binary fission. Binary fission is the asexual mode of reproduction. During binary fission, genomic DNA undergo replicate, and the original cell is divided into two identical cells. It means these are the bacterial cell, and the genomic DNA present inside the bacterial cell in the form of circular ring. And when binary fission occurs, this genomic DNA is divided into to form two layers, two bacterial cell, and this outer layer is called cell envelope. And this genomic DNA is divided and form two separate DNA structure, and these are the two separate cell wall, and these are form separate bacterial cell. These all are the process of binary fission. And first of all, binary fission begins when DNA replicates, followed by DNA segregation, division, and site selection, invagination of the cell envelope, and synthesis of new cell wall, which are tightly controlled by cellular protein. The new cell wall synthesis is tightly controlled by cellular protein. A key component of this division is the protein FTSZ. Means FTSZ are the key component of this division, which assemble into ring-like structure. Means FTSZ is assembled in ring-like structure. Other component of division apparatus then assemble at the FTSZ. Other components of division apparatus assemble in the FTSZ, and this machinery positioned so that division spills the cytoplasm and does not damage DNA in the process. So that division is spilled and spill the cytoplasm, and in this process the DNA molecule does not damage. As division occurs, cytoplasm is cleaved. When division occurs, cytoplasm is separate and cleaved, and that in form of two layers. And the new cell wall is synthesized. New cell wall, you can see this new cell wall is synthesized. This is all about the reproduction. Next page I will detail about the process. Show you the structure of reproduction. Here is the structure of process of reproduction in bacterial cell. Again, I will explain. These are the FTSN ring-like structure present inside the cytoplasm of the bacterial cell, and it can divide. It help to divide the bacterial cell and form new cytoplasm. And in this process, DNA molecule can't destroy, and the two separate DNA molecule and two separate envelope are formed. And this one bacterial cell is grow and form cell enveloped DNA, and DNA will also grow, and this this again form FTSZ ring structure. You can see the diagram. So this can all about the structure of prokaryotic cell. I hope you can understand well. If you like this session, please like, share and subscribe. Thank you for watching.